Well, good evening, and let me add to Joshua's very warm welcome. Joshua is the administrator for Truth in Science, and I'm the secretary for Truth in Science. And so this is a very new thing for us to do something as a student forum, and we hope that you will find it beneficial both to your mind and to your heart as well. This is indeed a follow-up to the Truth in Science summer school that we had in September, and we had over 100 students come along on that occasion, which was most encouraging. And so we did say that we would have about 5 meetings this year as a student forum, and then possibly next year we'll move it on to a monthly basis. So let me introduce myself. I'm a pastor of a small independent Baptist church. I'm also a biochemist. I've been a biochemist for 35 years. And 14 of those years I spent in industry working with enzymes. And I was the head of a research and development company, a pharmaceutical company doing research and development. And I've also spent 10 years in teaching in public schools. So I've taught biology, chemistry, and particularly physics at A level. So tonight we're going to be looking at functional proteins, which we also call molecular machines. Let me start off by telling you about this book. This book is by a man called Michael Behe. It was written in 1986, and it has the subtitle, The Biochemical Challenge to Evolution. And I do believe that biochemistry is at the forefront of challenging the misconceptions of Darwinian evolution. And Behe focused very much on the bacterial flagellum, which is like a little tail, is embedded into the outer membrane of that bacteria. And here you can see an electron micrograph, and the distance there, you notice just there, 20 nanometers. So these are, here's a larger diagram of that. These are 45 nanometers in diameter at the base. And that means that if you were to line these up side by side, you would be able to get 20,000 of these little machines in a single millimeter. That's amazing, isn't it? So many machines, so small, incredibly small, and as we will see, or if you read the book actually, you will see that these are 99.9% efficient. Well, I'm going to be looking more not at bacterial cells, but at human cells, and we all have these, billions of them in our head, in our brain, and going to every part of the body. They're part of the nervous system, and this is a single neuron. And on the left-hand side, you will notice that there are dendrites. These are what connects to other neurons. So one neuron will connect to as many as 1,000 different neurons through the dendrites. So they communicate to one another, a network. And then you've got, of course, the cell body, which contains the nucleus, and all of the organelles. We're going to look at a few of those. And then you've got this long axon. So the cell body contains the nucleus, the ribosomes, the mitochondria, which contain this ATPase we're going to look at tonight, the Golgi apparatus as well. And then the axon, it contains these microtubules and these kinesin molecules, which we're going to look at in some detail, which look like motor proteins that walk along the microtubule, a bit like a tightrope walker. And it's amazing to see. We've got a little animation to show you that. And then, of course, you've got the axon terminal at the end. So let's consider for a moment the neuron cell body. As a eukaryotic cell, it has a distinct nucleus containing the genetic code DNA, with all the information that is needed to manufacture thousands of functional proteins such as ATP synthase and kinesin, and of course all of the neurotransmitters which are required at the axon terminal on a molecular level, which is far, far away. And here is the end of one of those axon terminals. You can see the vesicles containing the neurotransmitters, which are then released into the synaptic cleft and then dock or binding sites on the postsynaptic dendrite of another neuron. I gave a talk about 3 weeks ago on neuroscience, and they weren't particularly scientists that I was speaking to, so I tried to explain some of this in a very simple way. So I was quite near Dover, and so I said that the axon terminal is like the terminal of Dover, where the ferries cross over, and then you've got Calais in France on the other side. In between, you've got the English Channel. We call this the synaptic cleft in a molecular level. And then you've got the minibuses, which are the vesicles containing all the swimmers, and the swimmers are the neurotransmitters themselves, which diffuse rather than swim across the synaptic cleft to the dendrite on the other side. So an axon potential travels at 270 miles per hour. It's not electricity that's flowing, it's actually the flow of sodium ions. But as they reach the axon terminal, it opens certain gates, which are called voltage-gated calcium channels. This allows calcium to flow into the presynaptic neuron, and they signal the neurotransmitter to vesicles to dock with the terminal and to release the neurotransmitters via exocytosis. These diffuse across and then bind to the receptors of the dendrite, and the signal is initiated in the postsynaptic cell. So these neurotransmitters, they're made of protein, and therefore they must be, first of all, manufactured. They're manufactured in the cell body. And you can see the nucleus there of the cell body, and this is surrounded by something called rough endoplasmic reticulum, which contain thousands of little ribosomes. In fact, there's about something like 5 to 10 million ribosomes in a single cell. And then, of course, they need to be packaged. This is by the Golgi apparatus, and these little vesicles containing the proteins within. And then they need to be transported from the cell body along the axon all the way to the axon terminal. And, of course, the whole process requires chemical energy in a form of adenosine triphosphate, or ATP. And this is the chemical energy that all living systems, including plants and bacteria, use for its chemical energy, a little packet of chemical energy. Well, of course, from your O-level biology, you will know that DNA, which is found in the nucleus, is transcribed into a single strand of RNA, or messenger RNA. This leaves the nucleus through the pore membrane and into the cytoplasm, where it meets these little machines called ribosomes, and they are involved in translating that genetic code into protein. So you have 3 bases in the messenger RNA. This is called a codon, and they are very specific for an amino acid, which are then brought in to the ribosome by these little taxis we call transfer RNA. I like to call them taxi RNAs, and each one picks up a particular protein, amino acid, and brings it into the site. And so the amino acids get joined together, and they form the protein, which is then folded in another machine and transported, no doubt by kinesin, to its required location. So it's a little bit like these ribosomes. Let's just talk about those as our first machine. They're a little bit like a tape recorder. And the tape recorder, a miniature tape recorder. The messenger RNA is a little bit like that magnetic tape, which contains information. And that information is then, as it passes through the tape head, the information is read and processed. And so this looks to me very much like a machine, a miniature tape recording. And what's amazing here, that these are only 20 millionths of a millimetre. That's 20 nanometers in size. So wonderful machines, very efficient, do a very specific job within the cell. And so here is a little animation. You can see the transfer RNA bringing in the particular amino acid into a binding site. The messenger RNA moves through the ribosome, like the tape through a tape head, and the amino acids get joined together to make the protein. But here again, these protein machines, these ones 20 nanometers, so small that if you were to line them up side by side, you can get 50,000 of them would measure 1 millimeter. And so, of course, then you've got 5 to 10 million ribosomes within that folded membrane, the endoplasmic reticulum. And it's called rough because it's got the ribosomes in there. And they are involved in the making all of the proteins that are needed by the cell. And they secrete those or release them into the cellular matrix of the cell body, including the neurotransmitters that we're thinking of. These neurotransmitters are then packaged by the Golgi apparatus, which then bud off as secretory vesicles. And they then need to be taken down, packaged up, and transported. And this is really brings us on to our second molecular machine. The neurotransmitters within the vesicles are physically transported along the axon. Remember, the axon contains the signal, the neural signal, at 270 miles an hour. But these kinesin molecules, they're walking proteins, they go much slower, 1 centimetre per hour. And so if you've got a neuron that's several centimetres long, it can take several hours for the neurotransmitter package to arrive at the axon terminal. And these amazing protein machines called kinesin. So you've got the kinesin here. This is a motor protein. We will see in a moment that it's fueled by ATP, and they literally walk along the self-assembling microtubule, which is again made of protein. And so they walk along 1 foot after the other. And so here is an animation that comes from the Discovery Institute of a kinesin, which is a protein that has 2 globular heads, or 2 feet on one end. One of them temporarily binds to the microtubule, and the other binds to ATP. And so here you can see it walking along. This is actually what happens. 8 nanometers every step, 130 steps every second. So when ATP is bound to the foot or globular head, it undergoes what's called a conformational change, and it flips over. And as it does so, it attaches to the second foot, binding to another ATP, which again flips over, and as a result it walks. And this happens in 10 milliseconds, effectively making 100 steps per second along the axon, travelling 8 nanometers a step, 1 centimetre an hour. And as you can see there, the other end of the protein, the neck, binds to the vesicle which contains its cargo, the neurotransmitter inside the vesicle. And so another protein, another motor protein, kinesin is one. They move away from the nucleus, and the dynein is another motor protein which moves back toward the nucleus and taking any spent material. So this is happening along the axon of the thing. So the question is, And it really brings us to our third molecular machine that we're thinking of tonight, and that is, where does the energy molecule, ATP, which drives these motors, like the fuel, the chemical fuel, where does this ATP come from? Obviously, we need quite a lot. ATP is involved in those ribosomes. Every amino acid that's joined together requires ATP. And proteins, as you know, can be several hundred amino acids long. So several hundred ATP molecules. So lots of requirement for ATP. Where does it come from? Well, they are actually found, they're manufactured inside something called mitochondria. These are some of the larger organelles. I think they might just appear as little dots in a light microscope, but you can see them quite clearly with an electron microscope. And we know that there is a very large folding within. So you can see in the matrix there, you've got these bound proteins, including something called ATP synthase. So 90% of metabolism is part of what we call oxidative phosphorylation. And this requires oxygen, so this is the aerobic side of respiration, and it also phosphorylates. It brings ADP and phosphorus together to make ATP. So the folded membrane contains enzymes, and these are part of an electron chain, which we're going to see in a moment, and they cache in energy from molecules such as NADH and FADH2. And we will see how they're made, because in the Krebs cycle, or the citric acid cycle, you can see it goes around, it comes in at acetyl-CoA, citrate into isocitrate. Let's just think about from isocitrate to alpha-ketoglutarate, just as one example. So there is this, I'm just counting up the carbons here, for a 6-carbon sugar. And then you've got the NADP plus, the NAD plus. This is reduced at the same time as the isocitrate is oxidized. And so it becomes alpha-ketoglutarate, and carbon dioxide comes off. So this is what we call a redox reaction. A redox reaction. So as the isocitrate is oxidized, the NAD is reduced, and it gains hydrogen ions. It becomes NADH. Now, what happens to the NADH and the FADH2? As you can see, this is a part of the membrane of the mitochondria, and it's got a number of proteins bound into that mitochondrial membrane. And so, from the inner mitochondrial membrane, in the inside, you've got the NADH, and this releases one of the hydrogen ions, and it moves through the molecule and is pushed out into the intermembrane space. So as you can see in the orange part of this diagram, many hydrogen ions. So you've now got a proton gradient, and so this is where it pushes it across, increasing the proton gradient in the intermembrane space. And then this couples on the last part of those molecules, the ATP synthase. And as you can see here, the hydrogen ions go downhill against the gradient. And as it does so, there is it causes the ATP to rotate and form ATP, the ATP synthase to rotate. So as a result of all of this, ATP is produced. It's the biochemical that fuels all energy-dependent reactions. So just take it again, the ATP synthase, you can see it on the right-hand side here in light green. This is the end set of a whole number of membrane-bound proteins, which we call the electron transport chain. And so stage one is the protons are pushed into the intermembrane space, causing this gradient potential, as you can see there on the left-hand side. And then as stage two, right at the other end, it flows downwards through the ATP synthase, causing it to rotate and make ATP. It's a little bit like one of those power stations, a hydroelectric power station. There you've got all this great potential energy in the water. As it moves down against the gradient, it turns a turbine, and that turbine then turns a generator, which makes the electricity in this step-up transformer. So that's how a hydroelectric power station works. And this is just the flow of protons down, and it causes the ATP synthase to rotate. So let me show you now an animation. This is the most amazing animation. This again comes from the Discovery Institute, showing this amazing rotating ATP synthase protein, which is embedded within the mitochondrial membrane.
It's been called one of the wonders of the molecular world, an amazing nanoscale machine. ATP synthase is a high-tech micromolecular power generator inside the cells of your body. It generates adenosine triphosphate, or ATP, an energy molecule that provides fuel that every one of your cells needs to function. Without this fuel, your cells will cease operation, and so will you. ATP synthase works like a rotary engine. The barrel-shaped rotator is composed of 10 to 15 protein parts called subunits. The rotator spins around, transmitting mechanical energy into the drive shaft of the machine, which helps make ATP. This drive shaft has a specially placed bump that opens and closes parts as the drive shaft spins around. This bump opens special protein subunits on the bottom of the machine. When the bottom subunits open, a spent energy molecule called adenosine diphosphate, or ADP, enters the machine. The mechanical motion causes the ADP to bind with an additional phosphate group, creating the ATP energy molecule. And the ATP drifts off into the cell, ready to power some biomechanical reaction. The ATP synthase machine has many parts we recognize from human-designed technology: a rotor, a stator, a drive shaft, and other basic components of a rotary engine. The ATP synthase is one of thousands of elegantly designed molecular machines inside your cells that make your life and all known life possible.
Wow, isn't that just amazing? And just to remind you, you have 100,000 mitochondria inside every single cell, each mitochondria containing 50,000 of those ATP synthase proteins. And for a full 360-degree rotation, it's very slow in the animation, obviously, but these travel 130 times every second. That's 7,800 RPM. And so from one glucose molecule, we get, it actually makes 38 ATP molecules, but 2 ATPs are used within the process itself. So really what I have to say as I come to a conclusion is, do machines such as ATP synthase, the ribosome we've looked at the kinesin, the walking molecule, do these machines, which are so efficient and have such a precise function, precision engineering, do they come about by chance? Or is it reasonable to say that they are designed, that they are made in a particular way from a designer? And so here again is the bacterial flagellum with all of the different parts. Michael Behe, the professor in America, said it reminds him very much of an outboard motor. And this actually uses protons as well from the outside of the bacterial cell wall to come onto the inside of the cell, and it drives these things around. It goes around 100,000 RPM, And that's just amazing. And it's 99% efficient. So his conclusion was that any machine that is 99.9% efficient must be, in his own words, and he's not a Christian, but he says that they must be designed. And this is very much what we feel as well. So just one other machine I'd like to talk about, and that's a machine that you may have, not inside your cell, billions upon billions of machines that keep you alive, but to think of another machine you might have in your pocket. What might that be? Well, it might be something like a mobile phone. And just one thing about a mobile phone, just to make you think, for it to have any use at all, there must be certain things that are designed by engineers that put it into such a small thing. It must have a microphone to pick up the voice and convert that sound into voltage fluctuations. And then, of course, it's got an antenna. It needs to transmit the information into the cell station somewhere else, antenna, or even a satellite. And then, of course, there must be the processor that picks up the voice and all the other things that go on. They're like mini computers, aren't they? And then, of course, all of this, like ATP needed for the machines, there needs to be an electrical supply in the form of a battery. And so we call this an irreducible complexity. And that means all of those parts need to be present and working for your mobile phone to work. We could think of a similar thing of a car engine. And none of it works unless all of it works. And so again, if you've broken your mobile phone, they may fix one part to get it going again. We call this irreducible complexity. And all complex machines do need, as something we know in science through technology, they need software for them to operate. And so computers, you've got the hardware, the computer itself, but then you've also got the software that runs the computer, both being required. Well, in a similar way, in biology, DNA is the software. It's much more advanced than the binary code that computers use. It uses these 4 base pairs. And you remember we looked at those little codons which code for an amino acid. Wonderful thing, great discovery made by Watson and Crick in 1953. But that's the DNA, is the code, the software. And the functional proteins, the molecular machines, are themselves the hardware. Well, if this is true, then who wrote the software, the DNA sequences, let alone manufactured the hardware? And to note just this, that for any complex system to function, all of the code and all the hardware needs to be placed together and available from the very start. And this is a great problem to those who believe in evolution. And so these are just some of the 3 molecular machines that we've looked at tonight, which are found in our cells, millions upon billions of them, and they themselves keep us alive. They're all part of an irreducible, complex system. Each one is needed for it to function. And so my evaluation is that each of these are designed by God and is evidence of his handiwork. As I will read in Romans chapter 1 and verse 20. For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they, that is us, are without excuse. Well, that's all I have to say. I've come within my 30 minutes that was given to me by Joshua, so I'm quite pleased about that. I hope I haven't gone too fast, and maybe you may have some questions. So I'll leave it there and hand back over to Joshua for any questions.